Hello again. So in this lecture, we will look at a few different things. We've looked at the central dogma. Now we'll look at a few aspects of microscopy, and then we will look at structures of bacterial cells and how they are shaped overall. So let's get into microscopy. Let's see what we're going to see. So the basic idea is that microorganisms are very small. If you look on this graph at the y-axis, you'll see each line goes down by a factor of 10. Humans are roughly 1 to 2 meters tall. Frog eggs are 1/1000th of that. Most bacteria are 1/1000th of a frog egg, so they're 1,000,000 times smaller than us, roughly. And that means we can't see them without without magnification. So, we have to use microscopes. And what we use in the lab are compound light microscopes. That means they have multiple lenses. They have 2 lenses. They have an objective lens and an ocular lens. The objective lens sticks down and gets close to the slide, and the ocular lens is what we look through. Both of those magnify the image a little bit, to different degrees. If you had a non-compound microscope, that would be a single lens, so that'd be a magnifying glass. So this is what we'll use in lab. This is really what I want you to think about. So what I've been talking about is magnification, and magnification is the act of making an image bigger. And that's obviously what we need to do. We need to make a invisibly small bacterial cell big enough to see it. It turns out magnification is easy to achieve optically. You can just stack lenses and magnify something a whole lot. You could take a picture with a great digital camera and just blow it up a lot, and that's another way of magnifying something. If you've ever done that, you realize, though, that you eventually reach a limit where you're just blowing up big pixels, and there's no more detail you can get. And what that's getting at is resolution or resolving power. And so resolution or resolving power is the ability to see that 2 separate objects are actually separate. Let's think about resolution. Typically, when people talk about resolution or resolving power, they're talking about the ability to tell that 2 objects are separate. So the 2 objects we're looking at here are 2 little dots, and we have adequate resolution to tell that they are separate. If we had resolution that wasn't quite as good, what we would see instead would be kind of 2 gray blurs, and they'd be bigger and they'd be closer together. Still, we can tell that they're separate, but if we make the resolution even worse, what we would see is something like this, just a blob. And so, so we have to think about how good is our resolution? How good is our resolving power? Can we see the difference between, can we see that 2 small objects are separate? And realistically, that's not what we're looking at in the lab. We're not looking at little dots. We're trying to see details in bacterial cells or in fungal cells. And if our resolution is not very good, we'll still be able to see that 2 cells are separate from each other, but we won't be able to see any detail. And that really is what we get with light. So the visible light can only give us a certain amount of resolving power. And it doesn't matter how well you build your microscope or how much it magnifies, you'll never see more detail in a bacterial cell than this. So this is a picture of 4 bacterial cells. You can see no detail. There's nothing we can see in them beyond their basic shape. And even their basic shape is fuzzy. where does this cell start and end? You can't really pinpoint it because it's a little bit blurred. And that's because the resolution of this image really is not so far different from the size of one of these cells. So that's something we're going to see. So one thing I want to show you is from astronomy, where this is much more like easy to see. And I want to show you the difference between two different space telescopes. The Spitzer Space Telescope was, maybe is, I don't know, about 1 meter across. It has something like a 1 meter mirror, and it takes infrared pictures of the sky. And it has similar magnification to the James Webb Space Telescope. But it turns out for telescopes, the resolving power is determined completely by how big the scope is, by how big that mirror is. The bigger that mirror is, the better resolving power you can get, and that's just an optical limit. There is no getting around it. There's no way to get better resolution without making a bigger mirror. So that's what they did. They made Webb, which is huge, and that this just blows me away to see the difference between these two. But so here you see Spitzer, and that's what you get with a much bigger telescope. It's the same magnification, but much, much, much better resolving power. So, for example, these 3 dots here, I don't know if they're stars or galaxies. How would I know? I don't know what they are. Over here, they're a blob. You can't tell that they're separate from each other because the resolution is too big, too coarse, too bad to show us that detail. And it also means lots of these little faint things just disappear completely into the noise. And that's really what we're dealing with when we look at bacteria under the microscope. We are stuck with the optical wavelengths of light, giving us a limit to our resolving power. If you have access to an electron microscope, there you're limited by the wavelength of electrons, which is much, much smaller, and so you get much better resolution and you can actually see details. But there you have to prepare samples in a very particular way. That brings us to contrast. Contrast is what lets you see the difference between the cell and the background, or between 2 different things in a cell. And most of the time, what we are going to see in our class is stains. And if you've ever, and that's what gives the cells their color, because most cells don't have any color. There's nothing to see. They are clear. They are colorless. So you have to stain them with some pigment that makes them visible. And if you've ever done histology and looked at tissue samples, they have been stained, usually to make the nuclei blue and the proteins red or something like that. Well, if you didn't stain them first, it would all be kind of grayish and very hard to see it, and you couldn't see the nuclei at all. So contrast is something we have to think about. If you ever use an electron microscope, you'll see you have to embed your samples in metal, or maybe you have to remove all the water and coat them with metal. That changes the shape of the cells. So electron microscopes are great. They give you pictures like this. This one is a scanning electron microscope. It shows you the surface features, but you can't trust them because this cell, to take this picture, they had to remove all the water from it by soaking it in different solutions. Then they had to put it under a strong vacuum and coat the outside of it with a thin layer of metal. And that definitely changes the surface structures. So who knows what these little hair things should look like? We don't know. But again, that's what you have to do to get enough contrast to see anything. You have to do something to give the cells contrast. And for this kind of electron microscopy, that means you have to coat them with metal. What we do in our lab is use prepared slides or stain slides. So one of the tasks you're going to have in this class is learning the parts of a microscope. So Here's a list of microscope parts and their functions, so what they do. This is going to be a little easier for you to learn than some of the other things because you're going to use these in the lab. But I urge you to learn this terminology. And go to the study guide to see how much detail. I think it will tell you exactly what you need to know about these things. And here is a handy diagram. There's also one in your lab manual. So what else? How do we calculate the magnification of one of these microscopes? Well, you multiply the magnification generated by the objective lens by the magnification generated by the ocular lens. Multiply those to get the total magnification. And again, 2 lenses, you could add a 3rd lens to get even more magnification, but you wouldn't get any more detail because we're already at the limit of our resolving power. And cool, that is... Well, that's all I really have in these slides for microscopy. I just want to point out one other thing. What are we looking at here? Why are they dark against a light background? This is actually a picture taken from a microscope that generates its own contrast. It uses the difference in refraction between the water and the cells. So light is refracted a little bit differently through the cells, and it blocks light that has come through cells, and it makes cells dark against a light background. So that's another trick to give contrast. And in the lab, we have a few microscopes that can do that, but mostly we're going to use stains to generate contrast. Okay, cool. I'll see you at the next video.